Hi, everyone, and welcome to the Pharmaceutical Clinician, bridging the gap between clinical medicine and the pharmaceutical industry. Now, I'm very excited for our webinar today to introduce a very special guest. We have Bassam Nasreddine Karout, who is currently based in Cambridge and has well over a decade of experience in the pharmaceutical space and also the diagnostic space. So his skill set focuses not only on the diagnosis, drug discovery, but also taking pharmaceutical agents to market. He's also worked across Europe, in Spain and Scandinavia. So Bassam, it's a pleasure to have you here today. Welcome to the Pharmaceutical Clinician.
Thank you for having us. It's good to be here.
Yes.
I appreciate a lot what you're doing. And I think doctors should be much more involved in the development.
Fantastic. So looking forward to your educational webinar today, sir.
Thank you very much. I would like to discuss today a very simple but powerful topic, which is how easy it is to make new treatments for life-threatening chronic complications, as long as you're operating outside of the small, narrow box of big pharma. Because the real barriers are not technical barriers. So a patentability barrier is important for a pharma company. It's not important for someone that wants to develop a product. Same as scalability. Many doctors would like to develop a treatment for their patients in a specific country or a specific region, and they don't care about the scalability of the product globally. And the minute you make peace with this idea, then treatment discovery becomes a very linear effort. You just put in the work and it works. It doesn't require any problems. And it's fun and it's very rewarding because you would be picking up what the big pharma has left untackled. So the patients that receive such treatments end up being very grateful and very, very happy. Let's imagine this. You're a large pharmaceutical company that would like to develop treatment for a life-threatening chronic disease. You get your brightest people together, and they come up with 60 solutions. From these 60 solutions, then we go through the patentability filter. And then those 60 become 30. Then we go through the scalability filter. And the remaining 30 become now 10. Then from these 10, we look commercially at which of these developments fit our existing promotional structure so that we don't have to invest in a new promotional structure. And then you end up maybe with 4 potential options. And this situation forces us to ask 2 very powerful questions. 1, are these 4 options anywhere close to being the best or most suitable for the patient? And 2, how massive is the market that is being untackled by the narrow scope of big pharma? And this is what I would like to talk about. My first experience with this was when I was a kid, and I worked with a very creative doctor, both MD and PhD, so he was a scientist and a clinician, ophthalmologist, Dr. Maciek Osenka from Poland. He's very creative and very empathetic person. And like myself, he believed that the only rules we need to adhere to are the rules that are set by physicians and by health authorities, not the rules set by industry. And they're very different. So patenting a technology is an industry barrier. It's not a technical or therapeutic barrier.
Makes sense, yeah.
So his approach was extremely simplistic, very powerful, and very linear. As an ophthalmologist interested in diabetic ophthalmology, he would go around Poland, asking ophthalmologists a very simple question. What do you face in your clinic that does not have a solution today that you as a clinician believe that there must be a solution? It's a very simple question. Then they would give him the feedback. Then we would go and repurpose an existing technology, or if we must, we'd create a new technology, and he would test it in vitro only. So he would only do the lab tests in a reputed university, then go back to the doctors that requested these solutions and say, look, I have found these solutions, I have tested them in lab. Would you like to do the in vivo testing? Then the physicians who have the patients that are suffering from the lack of the solution would offer to do the in vivo testing. And Masiek would then present these studies in international ophthalmology congresses. That was the model, extremely linear. What are you missing? He would create it, do a lab test, then come back and offer for the doctors to do the in vivo testing. Diabetic retinopathy was the one request that came from everyone. This is in 2010. And the reason was that the ophthalmologist would receive the diabetic retinopathy patient once vascular damage had happened. So now the ophthalmologist had to deliver the bad news and had no solution. There was no solution to offer. And it was the first time they met this person. And it was a very irritating position. So Masiek looked at what happened in the retina before vascular damage happens. And there was a threshold of nerves, of retinal nerves that had to be lost before any vascular damage would happen. So he built a very simplistic protocol of measuring loss of nerves in the retina. And because he had to be able to reach enough doctors to make it worthwhile financially, he kept the testing extremely low tech. So he tested the function of the retina, color sensitivity, contrast sensitivity, and acuity. And extremely simple. Now we needed a product. So now he went again to the doctors, but this time to neurologists. And he asked them, what treatment do you give for pre-amputations, for peripheral neuropathy? And they all agreed on 600 milligrams of alpha-lipoic acid as a drug. It's cheap, available, easy, stable. But because his interest is going to market fast, what he did was he created an alternative to this product with 300 milligrams, allowing it to be registered as a food supplement.
Fantastic.
He hit the market in 3 months. Now he had a product that's available. He had the technology that the doctors could use to detect diabetic retinopathy earlier. And when this product was delivered orally, the doctors would measure the results, the patients would document their benefits, and this was published in different ophthalmic congresses internationally and journals and ophthalmic journals locally. More or less, this is the full picture. And of course, he would then add testing in lab. So then he would add measuring the thickness of the retinal layer and so on to give it the objective touch. But end result is patients were getting better vision in as short as 6 weeks. The reason being is the blood flow to the eye is 10 times more than the blood flow to the brain. So the inflammation is there fast. It's the fastest occurring neurodegeneration in diabetes. But it also meant the treatment got there very fast. And if you're seeing results in 6 weeks, you're going to continue taking the product. Now, this is the simple look at it, what everyone sees. But if you have diabetes, if you're living with diabetes, and diabetic retinopathy is the first detectable neurodegeneration, and you're given an oral neuroprotector this protects everything. It's not protecting, it's not strictly for the retina. Unfortunately, back then, we did not have the platform to measure its impact on cardiac or on CNS or on other applications. But we knew that if someone is taking neuroprotectors as early as getting diagnosed with diabetes, it have more benefits. And this is what got me on the current route that I am. It was a great experience. Maciek was always creative and fun and impactful. And the second product he developed was for flashes and floaters. And that represented 50% of the population above 40 of age. So the numbers were massive. And 36 products later, I don't know how many studies later, he sold the business. It wasn't patented. There was not a single product patented.
Interesting. Yeah.
And they were food supplements or medical devices. So pharma companies could have probably made a better version. He sold it happily. And I would like to add that 3 years after this started, the industry, the community changed the definition of diabetic retinopathy from a vascular complication to a neurovascular complication, which then flooded the market globally with other alpha-lipoic acids.
Certainly a very,
Why not?
no, don't worry, a very, very incredible and motivating story there, in fact.
Yeah,
One thing that I wanted to ask, though, is I think it's quite incredible, or maybe it's my naivety. Industry, particularly the pharmaceutical industry, it's a powerful beast. And obviously what your colleague managed to achieve, is very inspiring. What were there any steps that he and his team had to be aware of? There was no competition with industry. There was no sort of no tension between the two camps. Or yeah, I mean,
So
I
I
don't
would
know. Maybe because
say
I don't
this.
know enough, but
No, of course we did. And I prefer to be as blunt as possible and hope that
that's okay.
people that are listening are creative and should be able to manage this. Abir, I think there's two elements here that frame the situation of having the pharma as an enemy or not. One, the project was sold to a pharma company.
Okay.
One.
Makes sense.
But the promotion was done exclusively by doctors. There was no online promotion. There was zero bonuses to pharmacies. So the doctors would recommend the product. They couldn't prescribe it because it's not a pharmaceutical. But 100% of the sales were doctor recommended. And the only place you could hear about these products were in science congresses or publications. So it was hyper credibility. It was more credible than a medical rep approach. And I remember asking him once, I remember this like it was yesterday. I remember this. I was asking him, why are they trying to buy us? Why don't they just copy the product? And he shushed me, of course, because he's older and wiser. And his answer was because they're going to have a meeting about what to discuss in the next meeting of the meeting they want to have with us. And by the time they decide how to approach it, I would have launched 4 more products.
Okay, it makes sense. Yeah, no, thank you for sharing. Yeah.
So I'm glad and happy to be here. So thanks for your questions also. A few years later, I learned from this that the physicians and the patients will buy the product from the persons investing in the science. And patents are relevant only if you're far away from the patient. If you're close to the physician and the patient, it doesn't matter. My subjective opinion, obviously. The second thing I learned is that the prohibitive costs of clinical studies are exclusively for big pharma budgets. So if a doctor is offering to do a clinical study with another doctor, the costs they exchange between each other have nothing to do with the costs with the pharmaceutical industry. Again, another very important barrier was dismantled from my view. At least 30 studies were conducted, and Masiek didn't have a budget. So a few years later, I was working as I worked with pancreatine. Pancreatine is digestive enzymes made from extractions from pork pancreas.
This is a very timely discussion, in fact.
Yeah, yeah. And it's within your area. So the more you ask me, the easier it is for me. Pancreatine was created in the late 1800s. It's a very safe product. It's a very cheap product. And everyone uses it. And it's used for chronic digestive complications. Now, pancreatine started having shortages about, I would say, more than 10 years ago, definitely. But every year, the industry would inform the world that this is temporary and that next year it will be fixed.
Yeah.
And every year, it became worse consistently. It's a very consistent manner. I have my thoughts on why this is the case, but there's many elements. But one of the elements was because of the extraction process of pancreatine, it is impossible to get a consistent enzymatic activity. So if you take a pharmaceutical pancreatine, 10,000 units or 25,000 units, within the same box, you could have up to 70% variance in enzymatic activity. So you can't make generics because you need to do equivalence tests.
Yeah, sure.
In absence of equivalence tests, how are you going to have generics? There's more reasons. I'm sure there's more reasons, and this is my limited view. But I know this because I used it to my advantage, obviously. So I wanted to create a product that covered these shortages because I could see it every year. There's bigger market, bigger market, bigger market, bigger market. But I wanted our product to be a superior product. I did not want a Me Too product. I wanted a jump from amoxicillin to azithromycin. I really needed that to allow us, because we're competing with really, really big pharma when it comes to pancreatine. And unless our tech was multiple times better, I knew we weren't going to get anywhere. Unlike the project with Dr. Osenka of alpha-lipoic acid, in this case, there was an alternative. There was just shortages. So we decided to use fungal enzymes that are used in the food industry. And what we did was we moved the digestive aid from the intestine to the stomach. I'm gonna further develop on this. Pancreatine is only operative in the basic medium, so in the intestine, which means it doesn't work for dyspepsia. It's not relevant for gastroparesis. And if you have cystic fibrosis, if you're living with cystic fibrosis and have 2 degrees acidic, extra acidic intestine, then pancreatine was particularly ineffective for you. So what we did is we got digestive enzymes that have a wider pH activity. So we got different lipases that would operate from pH 2 to 8.
Okay.
3 types of proteases that would operate from pH 4 to, I don't know, to 8. And then each one would have a different peak activity. So some of them would have a peak activity in the acidic medium, some would have a peak activity in the basic medium, covering the entire digestive tract. And then we did a double encapsulation, as I'm a big fan of mechanical innovation. And the external capsule delivered digestive enzymes with a peak activity in the acidic medium in the stomach, and an internal capsule that was gastro-resistant would deliver digestive enzymes with peak activity in the basic medium in the intestine. And following my previous experience, first we tested it in vitro against pancreatine 10,000 without naming brands, and in Valencia Polytechnic University. The results were exceptional because we're starting the digestive aid 2 hours earlier. So it's not a fair competition anyway. And
I
then,
get you. Yeah.
well, it's a different approach. And mechanical innovation, in my experience, always wins because it's simple, everyone understands it.
The best innovations usually are, I agree, very, very simple as opposed to the overly complex ones. So yeah, very much parallel thinking there.
As we got more comfortable with how powerful the technology was, then we conducted an in vivo study on cystic fibrosis patients. Now, the reason we chose cystic fibrosis patients was because cystic fibrosis patients would consume, besides pancreatic cancer patients, would consume the highest amounts of pancreatine daily, so 20 to 40 pills a day, depending on the dosage. And they were some of the most severely affected patients. So the burden of the cystic fibrosis is. So we thought if we could demonstrate that we are a reliable alternative in cystic fibrosis patients, it'll be very easy to go to less severe complications. The results in cystic fibrosis where we were able to reduce lipase, this is in vivo. We were able to reduce lipase intake by 79%. And we're able to reduce the painful daily symptoms by 50%. It was incredible.
Yes. No, it's a good outcomes for sure.
Yeah. But again, I insist, it's because we cheated. We moved the treatment from the intestine to the stomach. So now the heavy lifting is happening in the stomach unnaturally. We're digesting lipids in the stomach. So when the chyme is arriving to the intestine for nutrients absorption, it's already well broken. The intestine can focus on nutrients absorption and regenerating itself, rather than also the digestion, which was the previous case. And even today, if you go to the Cystic Fibrosis Trust website or Pancreatic Cancer website, they have sections. Write to your MP about the shortages of pancreatine or hoard any pancreatine you can get from the pharmacies. It's mind-blowing how bad the situation is. And now I face the problem because my technology was around 5 times more expensive than pancreatine because it was double encapsulation.
Yes, there is a cost factor, isn't there?
Of course. And it was really disastrous because we couldn't sell it anywhere. I developed this product with a food supplement company called Aura Health in Madrid. And our product was not reimbursed. It was not a drug, so the doctors were reluctant on prescribing. And it was multiple times more expensive. So now I had a product that was multiple times more effective than pancreatine, but no market to sell it.
Because of the cost factor.
Because of the cost, reimbursement, because I'm aiming for chronic patients. I don't care about dyspepsia. And we switched strategy. So I decided to go to markets where neither products, neither pharmaceuticals nor food supplements are reimbursed, or to go to markets where food supplements are reimbursed. And we went to markets where a doctor is able to prescribe a food supplement, or to markets where there was no more pancreatine at all. Very exciting.
Yes.
Really exciting. Because we ended up unwillingly, not on purpose, but we ended up becoming specialized in markets that were small enough for big pharma to leave us alone, but big enough for them to be lucrative for us. So Saudi Arabia, Philippines, Vietnam,
Mhm.
Ukraine, 26 markets later, I sold the business last year.
Oh, fantastic.
And we sold the business mainly because the company I was collaborating with was interested to remain a wellness company. And I was only interested in clinical and chronic and prescription aspects. So we couldn't really continue down that route. And that was my experience with pancreatine alternative. Again, I'm just checking my notes because I can get excited
Quite a journey there, I think.
and lots of fun.
Yes, I think yes, definitely. I mean, I think what you're seriously alluding to here is really very exciting, particularly from a clinician's perspective, because most of the time, what is happening is that a novel therapy or agent, in your situation, you've talked about food supplements and things as one example, and then we are the deliverers, say as part of the trial, but actually you're involved in the development, the modifications, the tweaking, understanding the development side. And I think that that's what is lacking for many clinicians, although you did cite one example of a clinician at the start that was involved in that. So my question therefore is, if clinicians were keen on development uh, and discovery, what opportunities are there available? Do you have any recommendations, steps that they can take, or is it still a sort of closed arena? And if it is a closed arena, how would we get more involved?
Um, well, this then I will answer your question with my last part of my presentation, which is exactly that.
Oh wow, okay. Wasn't expecting. Good, perfect.
So I think breaking this taboo that you have to have something patentable, you have to have something scalable, and you have to have pharma budgets to create solutions. It's just not true. It's just not true. It's not. If you want to be a billionaire, yes. If you want to be a millionaire, no. And if you just want to make some money and have fun, it's extremely easy. An alpha lipoic acid of 600 milligrams and alpha lipoic acid in food supplement of 300 milligrams, the supplier is the same entity. It's a different paperwork. It's the same batch of alpha lipoic acid being manufactured. If the doctor and the patient, it's important, I think if the doctor and the patient are involved in the creation and you're still testing everything clinically, not commercially, then I think it's multiple times easier than everyone seems to think and say. And I stopped inventing a long time ago. And
Mhm.
now
Yes.
I get doctors on board and they do the inventing. I have a network of international distributors. So any
Okay, nice.
technology that's invented, I'm able to take it to, we're present in 30 plus markets. And I can help them scale it. But if the technology is needed, you would know as a clinician, not me,
Yes, of
and
course, yes.
definitely not the pharma company. So
We recognize the sort of clinical issues, what's missing,
yeah,
but often we lack the solution forming in pharmaceuticals at least, um, unless we are heavily twinned with, people like yourself, um, or,
mm,
lab spaces, so on and so forth.
well, you need the lab space. That I
Yes, definitely.
agree. But it's not as complex or difficult as the world seemed to paint it, is what I would.
Yeah, I think that's the thing. Because to me it seems like a hidden world.
Yes,
But I think it is becoming more and more transparent, and obviously through
correct.
discussions like this,
Yeah.
we can really break down silos. Certainly.
I would like to explain my last endeavor, and I would like to come back to your point quickly. Um, a few years back, I decided to create a network of gastroenterologists involved in research to merge my experience from diabetic ophthalmology and my experience on digestive in one. we teamed up with Professor Per Hellstrom, who is a global reference in gastroparesis or diabetic gastroparesis and upper digestive complications. He has a lifelong career in Karolinska and moved to Uppsala some years back because of, easier to chill in Uppsala. And he remains active clinician and pharmacologist and researcher. We teamed up to approach gastroparesis because it was very similar to diabetic retinopathy. It was only detected once it's too late, and it could be diagnosed or detected many years earlier if the approach was more creative. And this meant he teamed up with Professor Aspion Dreves, also a clinician, and probably a top, I would say, a reference gastroenterologist, pancreatologist globally. And both of them, so Uppsala University and Uppsala University Hospital and Aalborg University Hospital, created DCODI, Digestive Complications in Diabetes. And what they do is they got diagnosis questionnaires of celiac disease, of gastric emptying, and of PEI. The patient joins, they sign a consent form, they fill a medical file, and they cross the results of these 3 questionnaires, coming up with a better understanding. It's not a diagnosis. We can both understand this. And we also study loss of nerves in the retina and peripheral neuropathy. So we study enteric neurodegeneration, plus linking it to the fastest occurring and the slowest occurring. In our last publication, we were able to link retinal nerve fiber layer thickness to different organ damage in long-term type 1 diabetes. CNS, cardiac, enteric, and peripheral. So few years later, 3 years later, this was started 3 years ago. So 3 years later, and few publications later, now DCODI is running in Denmark with over 850 patients. Ongoing. So they keep coming back, they keep coming back, they keep coming back, they keep coming back. And we launched in Sweden a month or 2 ago. And now there is 5 Nordic universities involved. So Uppsala, Karolinska, Lund, Aalborg, and University of Oslo at different levels.
Yes, yes,
So
yes.
some are more interested in neurodegeneration, some are more interested in gastro, and so on. And this means that if I were to go back in time and take Masiek's treatment of positioning alpha-lipoic acid at the first instant of diagnosis of diabetes, now we're able to measure its impact on all the different neurovascular complications, turning something abstract, which is neuroprotection to avoid amputation, into something tangible that the patient and the doctor are measuring and filling the data themselves. And as my wife says, I can talk. So please hit me with some questions so I don't feel like I've...
No, I mean, I collaborate and speak with many experts like yourself, and actually, I'm going to say I'm completely blown away. So yeah, it's made me realize there's a lot of exciting stuff just talking to you. I feel that excitement, and I engage with your passion and interest. I think the main thing for me, as we build this platform, trying to continue this engagement between, clinicians and people in sort of the know pharmaceuticals wise is really, I think the gap is allowing clinicians to be more involved in the development phase. I think definitely there's value in offering novel therapies to patients and being involved in that way. But I think the added value is then how do we also get involved in the development? I think that's very exciting. And I think for viewers out there, certainly get in contact with Bassam. I will make his LinkedIn profile available. And, we shall continue these conversations, Bassam. I think certainly you've shown that the perception where industry is not approachable is not actually quite the case.
No.
And we have to sort of get rid of that mindset and start holding hands more between hospitals
And patients.
yes,
And patients.
certainly
They
yes,
know because they're living.
they're living the issue. Yeah, no, 100% true. And I think that often is certainly what I see. We're involving patients in terms of trial design, the trial process around consent, our communication. Have we explained everything in a way that they understand? But are we actually involving them in the discovery side?
Yeah, then two different questions, yeah?
Yes.
Two different
Fantastic.
So
Great.
for all of your network that is in the UK, the results of Decodi have been submitted last week
Mhm.
and accepted to Neurogastro
Oh, wow. Okay.
Congress in September or October in London.
Mm,
And Professor Hellstrom and his team will be presenting the results. I will be
fantastic. Yes.
there. And
It's a
I
big
hope,
conference.
yeah, I hope to see you there and be able to share your insights and questions to help us make our work more relevant.
Definitely. Fantastic. Well, thanks so much, Bassam. I appreciate how busy you are. And, certainly everyone out there, reach out to Bassam. Like I said, I'll make sure his LinkedIn profile is available. And it's been a pleasure from the pharmaceutical clinician. Thank you so much, Bassam.
Thank you very much for having me
Thank you. Take care.
today.
Bye-bye.
Bye-bye.